Today, I am joining you to discuss our Vitessce framework for integrative visualization of single-cell data, and I will also discuss some applications. So for those who are not familiar, we currently measure cellular properties using both imaging and sequencing-based technologies. And these can produce measurements at the level of spots, which are a few cells, to the single cell level, to the subcellular level, where we might observe properties of individual organelles or transcripts. And for the sequencing-based techniques, these have been made possible due to advancements in microfluidics around 2015, and they allow us to, once we sequence reads, then map them to the genome, we can also map these back to the cells from which they originated. And there are these large-scale atlas generation efforts like the Human Biomolecular Atlas Program, or HubMap, and as well as disease or organ-specific efforts such as the Kidney Precision Medicine Project, or KPMP, which are using many different single-cell technologies, including those which are spatially resolved, to generate these maps of for example, human organs. And there are many goals of this work, but some goals include those related to experimental design. So to answer questions like how many cells are needed to answer a particular biological question, or to understand what are the molecular and morphological features of different cell types, to understand how to order cells along a trajectory such as development, to understand the gene regulatory networks driving disease processes, to understand cell-cell interactions that are involved in disease, to understand cell types that are present across organs and which properties of those are shared versus which are organ-specific, and which cellular properties predict disease response or could be used to define disease subgroups, and then related more to drug development, which parts of a gene regulatory network can be targeted therapeutically, and what are some predicted therapy resistance mechanisms that might arise. So thinking about this more in a computer science context, so before the single cell types of experiments, there were these bulk experiments, and you can imagine in kind of a voting analogy that you would see the results in terms of kind of the overall proportions of votes going to each party. Well, in the single cell case, we have these cells that are that have these barcodes, and we also have many different modalities, or for example, transcriptomics, where we sequence RNA transcripts, or epigenomics. And one example here is to determine the accessible chromatin regions in each cell, as well as proteomics to understand protein abundance in each cell. And for these technologies, in the voting analogy, you can imagine we see the results with the identities of each voter. And then we have the spatial analogs of each of these technologies, where you can think of the results as also having the address of each voter along with their identity. And the data kind of in the bulk case would be a vector of gene counts, whereas in the single case, we have this additional cell dimension to the data. And in the spatially resolved case, we have these spatial coordinates. But there are challenges in analyzing this data. Not only are there these different techniques, but also the different instruments and different types of measured entities, as well as these different scales from the subcellular to the multicellular, all resulting in these kind of heterogeneous data sets. And the size of the data is also growing. So over time, you can see here on this plot, each point is a study that measured some number of cells, and then on the y-axis, you can see that number of cells per study is on a log scale. So the data is really increasing exponentially. And this is a diagram that kind of reiterates the heterogeneity. So you have these different types of observations, and you might have measured different features of these. And we would then observe these different matrices or not observe certain matrices, as well as the spatial information. And then through computational analysis, we can derive things like dimensionality reduction coordinates or cell type annotations or cell segmentations. And of course, we can start to analyze this data using statistics. So this is just a toy example showing we can take the mean or the standard deviation or different quartiles of the data. But another goal of atlasing, of the single cell atlasing efforts, is to understand how to represent this information. And this recent paper notes that this is still a challenge as of last year. So this paper writes, as the atlases increase in complexity, their visualization and interpretation also do. So workflows should therefore be adopted to ease interpretation and visualization of atlases spanning tissue resolutions and omics layers. So to kind of step back, we asked the question of how are these data sets currently being visualized in publications. And so we did a literature search for these and identified these 221 papers, and then we filtered these down based on some inclusion criteria and analyzed over 1,800 figures within these single cell atlas publications, and we created like an interactive browser to view the results. And you can see here that there were some clear trends in kind of what plot types are used, as well as the kind of like the attributes of those plots. So for example, this small multiples technique, which you can see here, is where you kind of repeat this instance of a visualization, and you kind of change one variable, like here, the gene that's being visualized on the scatter plot. And so over 700 figures were using this kind of technique, as well as this custom annotations and category where the author of the publication had it kind of annotated a figure with text or outlines or arrows to indicate some kind of a finding, which makes sense since these are publication figures where the authors are trying to communicate something to a reviewer or to the audience. And then you can see we also recorded kind of different types of plots, like scatter plots, or like more domain specific plots like dot plots. And you can see here that in this browser, you can easily kind of view these patterns, like the small multiples with, for example, the multiplex imaging type visualizations, or violin plots. And you can kind of filter the data to see this, or for example, volcano plots are more domain specific plot types. So then we can think about how to add interactivity, and we might add interactivity to get an overview of a data set, to zoom in to items of interest, to filter out uninteresting items, or to get details on demand, or to relate different items to each other, and then to undo or redo these interactions, or to extract subsets of the data. And we can do all this using computers, so using the mouse or keyboard or other types of interactions. And then you might ask if there are interactive tools for single cell data visualization, which there are. So you can see on the right these two tools, this Cell by Gene and UCSC Cell Browser. But kind of these tools are currently limited in terms of the visualization design. So you can see these revolve primarily around these scatter plots, or in the bottom case, there's also a heat map. And they can be challenging to deploy. So sometimes these require specialized server-side software or proprietary file formats, and that require some kind of data conversion. And due to the heterogeneous nature of the data, they can be challenging to configure, and they're kind of disconnected tools for different types of data. So you see here there's scatter plot viewers and genome browsers and image viewers that are all kind of standalone, which can prevent this integrative analysis of the data. So we address this problem by developing something called Vitessce, which stands for the Visual Integration Tool for Exploration of Spatial Single-Cell Experiments. And you can see here it's a modular framework. So if you have, for example, the spatial data or imaging data, you can include a spatial visualization, or if you have different dimensionality reduction results, you can include scatter plots for each of those, or for example, a heat map for a gene expression matrix. And Vitessce does not require server-side infrastructure, so it loads data from standard web file servers or cloud object storage systems. And it supports this coordination of entities across views. So if a user selects a gene or a cell type, that selection can be reflected in all the visualizations. And Vitessce supports these data analysis environments in Python and R. So in Python, the Jupyter notebook, or in R, there's support for RStudio, and also loading data from standard kind of single cell data structures like AND data, or for imaging data, it supports the OME-TIFF format. And some of the tools that are used to build this framework are DECGL for kind of large scale. It was originally designed for geospatial data, but we're using it here for this single cell spatial data, as well as the D3 and Vega Lite tools for the more kind of statistical visualizations, and then HiGlass, which our lab previously developed for genomic or kind of genome mapped visualizations, and this VIV approach that allows to scale to large multiplexed images. Any widget allows the usage of this in Python notebooks, and then we previously developed a new kind of method for the coordination of different visualization properties across the different interactive views. So this figure is just kind of showing that we designed this with the kind of knowing that there's this patchwork of different available data that you might have, the different matrices or derived analysis results. So this is just a demo of selecting a gene, and then that selection is reflected in all the other plots. Here, this shows the cluster selection being coordinated with the genome browser tracks, as well as the scatter plot as the user selects different clustering results. This is a demo of using Vitessce in a Jupyter notebook. So configuring with Python your data, and then adding different views to the configuration, and then laying out the views and rendering the widget in the notebook. And then here the Vitessce can be used just as it would kind of outside of a notebook, where you can change different properties and make different selections. So there are already some projects using Vitessce, which kind of can be categorized into like publication associated websites, where some author makes their data available on a website, or different data portals for the cell atlasing projects, or data processing pipelines, or some even commercial software that we've seen start to use Vitessce. This is a list of some data portals where it's currently used, and I won't go through all of these, but we have kind of this showcase page of documentation where you can explore these if you're interested. So I want to just highlight some applications where we're using Vitessce in the lab. So One is for computational pathology, which is aiming to use machine learning to derive this digital pathology knowledge. So visual assessment is still a key part of this workflow, as well as the integration of other omics data. And our collaborators in the Kidney Precision Medicine Project are using computational pathology to understand kidney biopsies. So for background, there's this process of segmentation where you identify different regions of an image, like here are these cell boundaries, which then enable you to derive some information or to understand, for example, which cell types are in the image based on the protein abundance within each cell boundary. But we can do this for other structures in the image as well, like these functional tissue units, like the glomerulus in the kidney, for example. So our collaborators have developed this pipeline for segmentation of functional tissue units in the kidney, and using this pipeline can segment things like glomeruli or tubules, as well as regions that are relevant to pathology. So this interstitial fibrosis and tubular atrophy, or IFTA regions, are one example. And so we developed a way to view these types of segmentations in Vitessce. So here we're turning on the tubule segmentation, and as the user zooms in, more data loads at higher resolution. If the user selects a feature, for example, this area feature, we see the larger tubules in yellow and then the smaller tubules in dark purple. If a user selects a different feature like this luminal fraction, the ratio of the tubular lumen to the tubular basement membrane, we see that is now mapped using this quantitative color map. And then the user can turn on, for example, the cortical interstitia and the interstitial fibrosis and tubular atrophy, or these IFTA regions, and then turn on the peritubular capillaries segmentation, as well as mapping this to a feature like the area of these peritubular capillaries. And we can turn off the image to try to see that the density of the peritubular capillaries is lower in this cortical IFTA region versus cortical non-IFTA regions. And you can kind of see that qualitatively as well on the image. And we've run this, or our collaborators have run this segmentation pipeline on many of these images, and these are available to view in the Kidney Precision Medicine Project data portal. So you can kind of turn on and off these different types of segmentations in the image. And I just next want to highlight a use case where the analysis and visualization are kind of happening iteratively. So here our collaborators developed this spatial query algorithm to identify sets of co-occurring cell types. So first, this method builds a KD tree to kind of spatially index the data. And then this also enables either radius-based or nearest neighbor-based queries. And then it builds a frequent pattern tree. And this enables kind of very fast queries, and which we can then visualize. And so this is here running in a Python notebook where this spatial query algorithm is implemented. And you can see on the bottom right that there's this new spatial query manager view, which enables the user to change the parameters of the method and then run, like execute the queries. Oh, sorry. I'll just start from the beginning here. So this is kind of a fast video. So here the cell type information is first being visualized as it would kind of normally with Vitessce. And then the user will click this Find Patterns button. So the spatial query method will run and identify that these two cell types like met the parameters. If a user selects one cell type, they can run this cell type of interest-based query where this cell type, all of the results will need to include this cell type. So it uses that cell type of interest as kind of the starting point for the queries. And then that showed that this query resulted in the set of 3 cell types. Next, this is a different use case where we're comparing data. So from HubMap, which has these non-disease, which is profiling these non-disease tissue samples. But one of the goals of HubMap was always to enable comparative analysis. So this is a quote from the original publication describing HubMap, where the author said that they expect this to facilitate these analyses and understand how cells and tissue structures vary across individuals throughout the lifespan and in the emergence of dysfunction and disease. So you can imagine comparing this to the kidney precision medicine data, which has these different kidney disease subtypes. We conducted interviews with biologists, clinicians, and pathologists from the Kidney Precision Medicine Project to build a visual comparison system. And we approached this both from the data processing direction and the kind of visualization, development of the visualizations to view those results. And one of the challenges here was also the kind of lack of established ways to store the results of these types of comparative analyses. So we developed a pipeline which runs kind of pretty well-established methods like differential gene expression tests or gene set enrichment analysis or cell type composition analysis. And then the key part is that this pipeline stores the metadata about which comparisons were executed in such a way that Vitessce can query that data and offer it to the user. And through the user interviews, we kind of identified these different scenarios, the more exploratory workflow where a user doesn't have a hypothesis in mind, as well as the more confirmatory workflow where the user has, for example, a biomarker of interest that they hypothesize has a different pattern of expression between, for example, the reference data and the acute kidney injury or some other disease subtype or between disease subtypes. So they might select those groups and then view that data. And as the user makes their selections or clears their selections, they can move between these different kind of scenarios. We also developed some new interactive visualizations for the results. So this is a tree map visualization to answer the question of, are the sample and cell type groups kind of relatively balanced, as well as this plot for cell type composition analysis results. So here in the darker bars, you see those are the statistically significant results, and in the lighter bars, those are the not significant results, and then the direction of the bars kind of, so on the left is cell types that are enriched in the reference, and on the bars going to the right are more enriched in this chronic kidney disease group. And here we developed this interactive volcano plot where if the user hasn't selected groups, the comparison is between the cell type and the rest of the, between one cell type and the rest of the cells, or if the user has selected a group, then it's kind of a within cell type comparison between the, for example, acute kidney injury and the reference groups. And then if the user clicks a gene, they would view the, for example, the dot or the violin plots to understand the pattern of expression across all the cell types and between the groups. And here this contour plot is showing the data with also the dimensionality reduction in the dimensionality reduction kind of space, but stratified here on the left and right between the chronic kidney disease and the reference or the two groups that the user has selected. So this is a demo where the user first has not made any selections, and then here they select this gene, which is differentially expressed in the podocyte cell type. And you can see here that that it's highly expressed in the podocyte, but no other cell type. And then as the user selects a group, so reference versus acute kidney injury, the data kind of stratifies by that sample grouping. And then here the user is selecting a different cell type annotation, and then selecting one cell type, so the adaptive proximal tubule cell type, and then selecting a differential gene expression result that was significant, and then viewing that in the other plots. Here, if the user selects something from the gene set enrichment plot, then different member genes of that gene set or pathway are shown in this dot plot, and then the user can select different individual genes from the dot plot. And then we conducted some preliminary user interviews to understand or to get feedback on this. So one was kind of at the design stage where we hadn't implemented anything yet, and this user reported that they would like to see different levels of cell type annotation and to understand whether there's sufficient data available to make the comparison that they are interested to make. And they reported that the main kind of use case they could see for this tool would be to select a biomarker and compare between the groups. And they overall noted that, or they said, I think that covers what you would want to look at as a biologist or a scientist who isn't necessarily adept at doing their own coding. So they noted that for any kind of more complex comparison, they would want to take the data and do that using kind of Python or R or kind of standard bioinformatics tools based via code. And then here at the implementation stage, we performed another interview. So this user noted that they would like to see more context about which analysis methods were used or different parameters, for example, as well as better indication of which data was filtered or how the data was filtered if it wasn't shown. And then here, this user noted sometimes being interested in a single gene across different cell types, or other times being interested in many genes, but maybe one or a small number of cell types. And overall, their feedback was positive, but we still need to evaluate this with more users to make it more kind of usable, as well as to integrate comparisons of spatial data or other data modalities. So this is kind of future work. So one idea would be to use this kind of small multiples approach, and there are tools from the visualization literature that we could explore to do this, to deploy the system as well. So to at least the users within the Kidney Precision Medicine project who are interested to view this data, as well as we can always have better integration with ontologies or knowledge graphs to help them validate different findings, as well as to integrate additional analysis methods. So this is thinking more about the spatial query example I showed. So that's just one algorithm that our collaborators developed, but there are many other types of spatial data analysis methods that we could work to integrate Vitessce with, as well as there's always a need for the improved scalability of these tools, like for this 10x Xenium data, which can be very large. So Vitessce currently doesn't really scale to data that large, but we plan to work on this, as well as some things like improved error handling or improving the R package. So a lot of my focus has been on the Python implementation, but we can bring some of those features into the R package as well. So here I just want to acknowledge the funding for this project, as well as my lab and other collaborators that also worked on this. And if you are interested to use Vitessce, you can install it using pip from in Python and we have some examples, example Jupyter notebooks, as well as if you run into any problems, you're always welcome to email or reach out, or we have some tips in the README here. And if you are interested to use Vitessce in other environments, we have this documentation website.